Hello everyone, this is your mentor and instructor for this 4 weeks international level MOOC on Bioinformatics Essential Databases, Sequence Alignment and Ethics, organized by Department of Microbiology, KK Wag Arts, Commerce Science and Computer Science College, Saraswati Nagar, Nashik, in collaboration with Min Bioinformatics Services. Now I welcome you all in the first module, and the part of first module, that is module 1.1 and 1.2 of the foundations of bioinformatics, and we'll start with the today's session. See, foundation of bioinformatics. In this particular module we'll learn about, and this is the week 1 module, and in this week 1 module we'll learn about computational biology, bioinformatics, what are the data types in the bioinformatics, and databases. So for this video, we'll learn module 1.1 and 1.2, that is the computational biology, bioinformatics, difference between them, and the what is the difference between in vitro and in silico experiments. And in next module, that is module 1.3, we'll learn about the data types and databases. See, in the science, there are three major fields: life sciences, provides molecular genetics, cell biology, genomics, and structural biology frameworks required to formulate biological questions. So basically, using this life sciences, we can create biology related questions. And in computer science, this computer science field will deliver algorithms, software engineering, database management, and cloud infrastructure for processing massive bio data sets. Now, then we'll take the help of another field, that is quantitative sciences. It applies probability, statistical modeling, machine learning, and biomathematics to derive the statistical confidence from noise. So, while learning this bioinformatics or computational biology, we require this a combination of the all these four sciences. Now, bioinformatics. What is the bioinformatics? Bioinformatics is the creation of tools, algorithms, software, and databases. Here we do information engineering, sequence alignment, data storage. Also, we manage, annotate, and analyze large-scale biological data sets. We works on the software platforms, annotate data set, databases, and analytical pipelines. And this bioinformatics is slightly different in compared to the computational biology. In computational biology, we work on the building computational models to answer biological queries. We do system modeling, dynamic simulations, mechanistic insights. We understand biological systems through algorithm modeling. So here includes theoretical frameworks, biological insight simulations, etc. So there is a fundamental difference between the bioinformatics and computational biology. Although these words are used widely in replacement of each other, but there is a little bit fundamentally they are little bit different. Now, what is the wet lab? That in vivo and in vitro things. So in vivo, we perform the assays using the live or inside the living organisms, like animal model trials, like working on the mice and working on the actual cells and everything. And in vitro is experiments conducted in control test tube or culture dishes outside living organism. Here we don't use the actual living organism, we use the test organisms or the cultures. So basically in vitro is everything we cover, we take up in our hand, we take up in our control, and then we perform the experiment but outside the living organism. And in vivo means we perform the experiment inside the living organism. So basically due to this in vivo and in vitro techniques, we got the high biological accuracy, but it's a resource intensive, it requires um, highly means it requires great amount of funding and everything, and time consuming. It's a little bit time consuming and ethically constrained. So there are many ethical issues. Actually, a very long process. Now, coming to the dry lab, in silico experiments. Now, in silico, biological experimentations conducted via computer algorithms and silicon based microprocessors. So, in short, silico. See, microprocessors which are used in the computers are made up of the silicon, right? So, in silico means, in short, in a very simple manner, we can say that in silico means inside the computer. So, whatever experiments if we conduct inside the computer, that are called as in silico experiments in simple words. Now, where we can apply these things? We apply these things in molecular locking, sequence alignment, virtual high throughput screening, and metabolic pathway modeling. Now, what are the benefits of this in silico modeling? High scalability, rapid through output means we can scale up this technique, or we can scale up the experiment to the great extent, and we can do the rapid throughput means generally within some time we get the high outputs from the experiment, and cost effective hypothesis generation means, see, for working in that in silico, working on in silico experiments doesn't require any kind of lab or anything, just one simple computer, that's it, you can do until and unless the computer has the that specific specifications, that's it. Now, how we can transform this modern R&D means, what is how these in silico experiments are transforming the modern R&D. So virtual drug screening, in silico docking filters millions of chemical compounds against target protein structures in days, replacing months of physical bench testing. See what happens during the in vitro or and in vivo things of the drug discovery, that requires working for months and months, years and years. But in virtual drug discovery, if we take the example of virtual drug discovery, we can do the same thing within the I say now here it is a days, but if your data is small, if your input is small, so you can take up the things within the hours also. So it requires days. We are just normalizing the things in days. So, and cost risk mitigation means prevents costly benchtop failures by eliminating non-viable candidates early in silico before entering preclinical trials. That is the very good benefit. And see what happens actually, if we are performing suppose if you are performing on 1,000 compounds for the drug discovery, and you are getting only, let's say, 10 to 20 compounds as the drug candidates. Now the remaining 990 to 980 compounds are not of use, means uh, we are just wasting our time, energy and money in doing unnecessary things. So to avoid that, see, once we work in the in silico, that time what we do, we performs the entire thing in the computer, and we get the results before starting the preclinical trials, means whatever best candidates that we are getting after processing 1,000 compounds in the lab, and processing of that 1,000 compounds, yes, also it will require about 2 to 3 months, right? So in silico we get these results in I will say within a one day we will get results for the 1,000 compounds, right? So we'll get that suitable 10 to 20 compounds within the day. So it will also, the process is very simple, and it doesn't cost anything, right? It just costs some amount of data, that's it, and data is very cheap nowadays, right? Likewise, we get the non-viable candidates early in silico before entering the preclinical trials. So that's it for the module 1.1 and module 1.2. We'll meet in the next module of the biological databases. It will be very short module, but then we'll have some experiments, or we will have some demonstrations of the actual visiting the databases, right? So till then, thank you, and please subscribe the channel, like this video, and if you have any doubt, just ping me in the WhatsApp group, or you can chat me in the my DM. My number is in the WhatsApp group contact. Thank you.